Hello everyone, welcome to the new session of Plant Growth and Development Part 1. So in this session we are going to study about the introduction to the plant growth and development, okay, and then we will also talk about the differentiation, redifferentiation and de-differentiation, right? And then we will also talk about the different forms of the growth. Okay, so let us start the session. So, you have already studied the organization of flowering plants in chapter 5. Okay, so chapter 5 we have studied about how the plants has been organized. Okay, we have studied about the morphology of the flowering plants in chapter 5. And have you ever thought about where and how the structures like roots, stem, leaves, flowers, fruits, and seeds arise, and that too in an orderly sequence? Okay, so this growth of the plants is well organized, right? And if we see any you can say seeds is germinating, then you see that the roots is coming out, the shoot system is coming out, right? The first leaves that emerges out from the seeds. Okay, so all you see, right? So have you ever thought about where and how the structures, like from where this roots is coming out, from where the stems is originating, from where the first leaves has been formed, okay, and how the flower, the fruits and seed arises in the plants, and how is this in a occur in a very sequence way, right? Like we never see that the formation of fruits before the formation of the flower. Okay, then first there is a flower, then there is a fruits, then there is a seed. So this is a very orderly sequence, the sequence of the things that is occur in a very order way. So have you ever thought about it, how is this occurring? You are by now aware of the term seed, seedlings, plantlet, mature plant. Okay, so these are the terms that you must be aware, that what is seeds, what is you can say the seedlings, what is a plantlet, and what is a mature plant, right? So the term seed, that is the ovule. After the fertilization process, the ovule changes to seed. Okay, and when you soak this seed in water, then what happened? Due to the warmth, due to the water availability, the seed germinate. Okay, and when it germinate, it give, first of all, it give rise to the radicle, and then comes out the plumule, right? So that form the seedlings, that form the seedling. When this seedling is established in the soil, okay, then the plant is there, the formation of the plant, it is having development of the root system, and the upper above the ground, then it is having the shoot system, right? And after due to the availability of the soil minerals, okay, due to the availability of the water, fresh air, the plant grow and forms in a mature plant. So you've also seen that trees continues to increase in height or girth over a period of time, right? So you must have seen this, that after 4 or 5 years, the plant which you have planted in your garden, after 4 or 5 years, it changes to in a form of a tree, right? It happens. Yes. So you can see the girth of or the width of the tree grows, and also the height of the tree. If you measure the girth or the height of the tree every year, you see that there is an increase in the height, there is an increase in the girth of the tree, right? So how this is occurring, how this phenomena is occurring? The plant which you have set in a very small form, now it becomes a mature tree. So how this is occurring, how this growth is occurring? So we are talking about the growth of plant, right, in this particular chapter. So let us see. However, the leaves, flowers, and fruits of same tree not only have a limited dimension but also appear and fall periodically and sometimes repeatedly. So not only we always see the growth, okay, we see the growth, yes, in a growing season they show, but there are some season in which they shed off their leaves, they shed off the fruits, flowers, okay, all the flowers, fruits and leaves, they get shed off from the trees. It happens. And so sometimes you see this is a very frequent, and but sometimes you see this is occurring in a periodically way, right? It is occurring in a periodic form. So why does vegetative phase precedes flowering in plant? Okay, after the plant get established, okay, the seedling is there, then it becomes a plantlet, and then it forms, you can say, a, a plant. And after a vegetative phase, a particular phase that is called as a vegetative phase, now there is a flowering phase in the plant. Means the flower comes out in the plant, and all plant organs are made up of varieties of tissue, and we all know that there are the different types of tissue. There is an meristematic tissue, there is a ground tissue, there is a permanent tissue, there is a complex tissue, right? So there are the different types of tissues in the plant. And is there any relationship between the structure of a cell, tissue, and organ and the function of they perform? So have you ever thought that there is some relation between the cells and the tissue? Okay, tissue and the organ, and how these organ perform the functions, right? So suppose if we talk about the leaves, so leaves are an organ that do perform the process of photosynthesis, right? They are the major organ that perform the process of photosynthesis because they are having a lot of chloroplast in them, in their cells. And if you see the structure, or you can see the arrangement of the cells and the tissue in the cell, it is very, very different from that of you see in the case of the stem and the roots, right? So there must be some relationship between the cells and the tissue. There are certain types of cells that is found only in the case of the leaves. There are certain types of cells that is only formed in the case of a stem. And there are certain types of cells that is only you see in the case of roots. So all these cells are not function individually. They unite and form tissue. And this tissue perform a particular function, right? So can the structure and the function of these be altered? Okay, altered means can you change these structure and the function? All cells of the plants are descendants of the zygotes. We all know that after the syngamy, there is a formation of zygote. The zygote divide and redivide, okay, and forms the embryo. And from the embryo, this whole plant is formed, right? So the descendants of all the types of tissue, all the types of organ is coming from where? It is coming from the zygote. It is a single cell. The question is then, why and how do they have different structural functional attributes? So from where only one cell is there, but how they are having so many different types of structures. So, and all these structures are performing the different functions. Like leaves is forming the different function, stem is forming having the different function, and as same roots are also having their different functions. But if you see, if you think about, then these organs like roots, stem, and root, stem, and leaves of the plant, they have been formed from which cell? Zygote, a single cell, right? So development is the sum of the two process, growth and differentiation, right? So it means that the zygote divide and redivide and form type of a particular type of cells. Now these cells, a particular type of cells, unite together and form a tissue. Different tissue unite together to form a organ. An organ perform a different functions, right? So there is two things. Okay, the development, if we say that the plant is in the development stage, so it means that development is the sum of two processes. Two different processes are there. What are those two different processes? Growth and differentiation. Growth and differentiation is there, right? So growth means the plant is in the growing stage. It is increasing its mass, right? It is increasing its size. That is the growth. Differentiation, the zygote divide and redivide and forms a number of cells. Now, out of those cells, some of the cells get differentiated into another type of cells. Okay, some of the cells get differentiated into the another type of cells, and they form a particular type of tissues. Now different tissues unite together and form a different organ. So the organ which is leaf, if you see, it is very much different from the stem, and the stem and leaves are very much different from the roots as well. So you see the difference in the organ, you see the difference in the function, you see the difference in the cells. There are number of cells. So how these cells are formed? These cells are formed from differentiation phenomena. Okay, so the development is the sum of the two processes you see here, that is the growth and differentiation. Okay. So to begin with, it is essential and sufficient to know that the development of a mature plant from a zygote, okay, zygote means fertilized egg, follow a precise and highly ordered succession of events. Okay, so if you talk about the development of the mature plant, how the mature plant has been formed, how the mature plant has been organized. So we say that the mature plant has been formed from one particular cell, only from one cell, and that is the zygote. And zygote is formed from the process of syngamy, that is the fusion of the male and female gamete. Okay, when the egg get fertilized, it is formation of the zygote. Now this zygote divide and forms the number of cells, and these the division of the cells, okay, it is a very precise and ordered succession of events. Means every step is pre-decided, that at which stage how many cells are formed, how what is the orientation of the division of the cells. Okay, everything is pre-decided. It is never happening in a random form. It is always in a precise and highly ordered succession of events. Okay, so during this process a complex body organization is formed that produces roots, leaves, branches, flowers, fruits and seeds, and eventually they die, right? So what happened here during the process of the formation of a complex body? Okay, what is the complex body here? The complex body is whole plant, how the plant has been formed. Okay, the complex body is the whole plant, the formation of the plant. Okay, so how this body of the plant has been organized, how they have formed the roots, how they have formed the leaves, the branches, fruits, okay, flowers and seeds, and eventually they die even, right? So this is the you can say that the they are in a very ordered and this following the very succession of events. Okay, so first of all, the seeds germinate. The seedling get established. Okay, the seedling get established in the soil. Then it forms a plantlet. You can say the plantlet, and then slowly and slowly the plant show its growth in height, in the width of the its stem, and after some years, it changes to what? It changes to a mature plant. Okay. Now, this mature plant is having a well developed root system, well developed shoot system. In the shoot system, you can see there is a one main stem, and from that, many different branches are emerging out. Now, these branches are having a well developed leaves. Okay, and there are different on the branches, you can see there are different flowers is emerging out, and after the pollination process, there the flower changes to fruits. Okay, the female parts of the flower, that is ovary, changes to fruits, right? And inside the fruits there are ovules, so that ovule get changes to seeds. So these are in a very ordered sequence, and also uh, after a certain duration of the time, the plants, the trees, they die also, right? So this is a very precise and highly ordered sequence of events if we see in the growth of the plants. So here you can see this is a seedling. Okay, this is a bean seed, and you can see that this is the soil layer. And when you sow a bean seed, you can see the first thing that is coming out. The first thing that is coming out is the radical part. That is the root. The root comes out and it gets established so that it can first the root comes out, root part. So it get established in the soil. It can absorbs the water. It can absorb the water which is available in the soil. So the water get absorbed and it reaches to the leaves. Okay, the leaves which is present here. So here you can see they are having the cotyledons. So cotyledons is also there. It comes out in the form of the first leaf, right? So here you can see this whole seedling part is came out of the soil. So here you can see the seed coat, that is the covering of the seed, it get emerges out. Okay, and you can see the cotyledon is there. So cotyledon changes to first leaf, and you can see the later some other leaves are also emerging out, and you can see there is an establishment and growth of the root as well as in deep soil. So the roots is going down and down, and it is also giving rise to many different branches. Okay, so the plant get established, and now here you can see the plant is having many branches also, right? So this is the germination and the seedling development in a bean seed. Now, the first step we see in the process of the plant growth is the seed germination. This is the first step that we see in the growth of the plant. Suppose the growth of the plant is occurring, then the first step that will occur, that is the germination of the seed. Unless there is a germination of a seed, then you cannot see the other steps. Okay, the other the process or the steps that you see in the case of plant. The seed germination when favorable conditions for the growth exist in the environment. Okay, seed only germinate when there is a, you can say, the favorable condition that exist for the growth of the plant. Okay, the environment that favors the growth of the plant with some of the factors, that is the availability of the water. The very, very important point for the germination of a seed, the seed must germinate when there is a water available. When the water is available, the seed germinate. Okay, the air is also the another factor. In the absence of air, the seed cannot germinate. Now, in the absence of such favorable condition, the seeds do not germinate and goes into the period of suspended growth or rest. So, when the favorable conditions are not available for the seeds, okay, the favorable conditions are not available for the seed, then of what happened, the seeds in is occurring the seeds sleeps in a form of like they are in the suspended growth or the resting stage, and that you can also say that is it is in a dormant stage. Okay, so that occur when there is an absence of such a conditions. Okay, the factors that is required for the growth of the germination is not available, then the seed is under the period of suspended growth or a rest, or you can also say that the seed is in the dormant stage. Okay, once the favorable condition return, the seeds resume its metabolic activities and growth take place. So this we see that this the resting stage, or you can say the dormant stage, is not for the long duration of time. Once the favorable condition is available to the seeds. The seed resumes its metabolic activities. It starts performing its all the metabolic activities and growth takes place. Okay, so the growth take place after the coming of the that is the favorable condition back to the seeds. Once the favorable conditions comes back to the seeds, or you can say it start the metabolic activities of the plant again get started, and the growth in the plant you can see. Now, in this chapter, you shall also study some of the factors which govern and control these developmental process. Okay. So we will see in this chapter what are the factors which are responsible and that control the developmental process in the plant. So these factors are both intrinsic, that is the internal factor, as well as the extrinsic to the plant, means from the outer, right? The factors that are responsible for the growth of the plant, those factors are two types. Intrinsic means within the plant, and extrinsic, that is coming from the outside. Okay. So external factor, if we talk about, there are so many external factors. So the external factor is the availability of water. Very, very important. If the water is not available, the seed will not germinate. Okay. So this is very, very important. Air and water. If we talk about the intrinsic factor, then there are some, you can say the enzyme get activated, and those enzymes help to solubilize the food material which are in there in the form of stored form in the seeds that is available now for the growing of the or growth of the embryo. And the second thing, there are certain hormones. Okay, there are certain hormones that are responsible for the germination of the seeds also. So that is also there. So these are some intrinsic factors that is working on the germination of the seeds. Okay, so both the factor must be there. The growth is regarded as one of the most fundamental and conspicuous characteristics of a living beings. Okay, so very, very important. It is one of the most fundamental and conspicuous characteristics of a living being, right? It is a very important factor. And without that means this is one of the fundamental you can say the fundamental factors that is or you can say the characteristics that always occur in the plants, and that is responsible for the growth of the plant. Okay, that is responsible for the growth of the plant. Now, what is growth then? What is growth? Growth can be defined as irreversible, permanent increase in size of an organ or its parts, or even of an individual cell. If we say that the growth is occurring, then the growth occur in a plant if there is a permanent increase in size. This increase in size is not reversible process, it is irreversible process. It means when once the growth occur, it never occur that the growth return to back to its original position. No, it never happen. So when there is a growth, it means that there is a permanent increase in size of an organ or any of its parts even. And you can say that there is an individual, you see that there is an individual growth in one particular cell, then also it is a sign of a growth. So this is the definition of the growth. The growth can be defined as an irreversible permanent increase in size of an organ, its parts, or even an individual cell. Okay, even in an individual cell. Generally, the growth is accompanied by the metabolic process. So how the growth is going on, how the growth is taking place in the plant or in any other organism, it is just because of the metabolic activities, metabolic processes that is occurring in the plant. And these metabolic activities are both the type, that is the anabolic as well as the catabolic. Anabolic means something is synthesized in the body. Okay, something is synthesized in the body, and that is called as anabolic process. And something that is broken down or the degraded, then it is called as a catabolic process, right? Catabolic process. So the growth is accompanied due to these metabolic processes, that is both anabolic and catabolic, and that occur at the expense of energy, right? So this metabolic activities is not occurring without any expense of energy. It is occurring due to the availability of energy. Therefore, for example, expansion of a leaf is a growth. The leaf growth, you see the size of the leaf, initially it is very small when it is emerging out, but slowly and slowly its size increases, the area of the leaf get increases. And this you can measure even. This you can measure even when the leaf is in new coming out. You can place that leaf without plucking out from the plant. You can place the leaf on a graph paper and you can draw the outer boundary and leave that leaf. Okay, it is still attached to the plant, and you can find the area from that graph paper of that small leaf. Okay, now after a week you see that the size of the leaf has started increasing and it become very mature leaf. Initially it is a new leaf that is emerged out. Now it becomes a you can say the permanent leaf, and it is increases that size, and again you just make a area of that leaf by placing it on this graph paper. Now you can see the area has been increased out, and you can find out what is the difference between these two area, right? So this is an expansion. This is showing the expansion of a leaf, and this is also a type of growth. This is also a growth. Now, how would you describe the swelling of a piece of wood when placed in water? When you place it in water, the wood get swells up. It gets swells up. Okay, this wood gets swells up, so you can say that it is a growth. It is a growth. But when you place that wood out of the water, then what will happen? It again, after some day, you see that it is coming to its original place. And sometime it happen also. The doors, the wooden doors, which get closed and open very easily. But what happened in the rainy season? Some of the doors, they get expand, they get water from the outer atmosphere. Okay, and they get swells up. Even sometimes the rain water also get on this door, and at that time they absorb the water and they swells up. So the door, the wooden door that was opened and closed earlier very easily, now it doesn't open easily. It doesn't open easily and it doesn't get closed easily. So this is just because of the swelling of the piece of wood. This is the swelling of the wood. And why it occur? Because it absorbs the water. Now, when the rainy season is gone, the water get evaporated from this wood. The door get open and closed easily. So it is coming back to its original position. And growth is defined as irreversible. It is irreversible, but here you can see the condition is irreversible. The size that has been increased, it is coming back to its original position. So this is not a growth. Plant growth generally is indeterminate, means it is not predetermined. Plant growth is a unique because plants retain the capacity for unlimited growth throughout their life. Now, plant is having one of the unique phenomena that it shows its growth throughout their life. In the case of plant, you see the growth throughout their life. Now, this ability of the plant is due to the presence of meristematic, a certain locations in their body. Okay, so they are having the meristematic tissue, and you see that the plant shed off their leaves, but when there is an season that favors the growth of the plant, then a new leaves emerges out. You see that, right? So this is the ability of the plant is due to the presence of meristematic tissue at certain locations in the body. So meristematic tissue are present at the certain location, that is at the apex of the plant, that is the shoot tip and root tip. Always there is a growth there. Okay, there is also growth in the intercalary meristem form, that it give rise to the new branches, and lateral meristem, that is the increase in the girth of the main stem, right? The product, however, soon lose the capacity to divide, and such cell makes up the plant body, right? So the cells of such meristem have the capacity to divide and self-perpetuate. So the cells of meristematum is having the capacity to divide and redivide, right? It is having the capacity to divide and redivide and self-perpetuate. Now the product, however, soon lose the capacity to divide. Whatever the cells has been divided of at the activity of the meristematic tissue, it sees its activity. It means that it loses it the capacity to divide, and such cells makes up the plant body. So the new cells which has been formed get differentiated. Some of the cells get involved in the process of formation of the leaf. Some will forms the new cells of the stem. Okay, some will form in the lateral meristematic tissue. It forms the you can say new xylem and phloem, outer side and inner side. So you can see there is an increase in the girth of the stem, right? So this occurs till the plant is alive. The growth you can see in the case of plant, okay, plant or tree, till the plant or the tree is alive. So this is a very unique thing that you see only in the case of plants, but not in the animals. Okay. So this form of growth where the new cells are always being added to the plant body by the activity of the meristem is called the open form of growth. Okay. So the plant is always having its meristematic activity. The plant is having the meristems. Okay. Every year you see that there is a shedding of the leaves and there is a coming of the leaves. New leaves are again emerging out. So how it is occurring? If once the leaf has been fallen and it is a very aged tree, then also you see when the season is coming back, okay, the season that favors the growth of the tree. So at that time you see that the new leaves are emerging out from the trees, right? Okay, and the new cells always being added to the plant body by the activity of the meristematic. So from where this new leaves are formed, new stems are new branching are from, because of the meristem, because of the meristematic tissue. And this is showing the open form of growth. What would happen if the meristem ceases to divide? Does this ever happen? So what will happen if there is this meristem which is showing always its division activity? It ceases its activity, ceases its division activity. Then what will happen? Then there you will not see any growth in the plant. Okay, so does this ever happen? So still the plant is alive, till the tree is alive, this never happens. The meristematic tissues are there and it always divide and redivide. So in earlier classes you have studied about the root apical meristem and the shoot apical meristem. Okay, the apical meristem, it is present at the root apex as well as at the shoot apex, right? So you know that they are responsible for the primary growth of the plant and principally contribute to the elongation of the plants along their axis. Right. So when you see the meristematic activity at both the apex, at the shoot apex and at the root apex, what you see in the case of root system, the root goes down and down because of its activity. It's having the root cap and that protect the apex of the root that goes down and down. Similarly, you can see in the case of shoot system also. The shoot system, you see there is an apical meristem at the apex, shoot apex, and it divide and redivide, and the plant is growing, okay, along their axis, right? So such type of things you see in the case of plant, and that is very, very unique things. So you also know that in the dicotyledonous plant and the gymnosperm, the lateral meristem, vascular cambium, and cork cambium appear later in size, right? In the case of plant, okay, dicotyledonous plant, and also the gymnosperms, the lateral meristem, that is the meristem, or you can say the cambium that is present in between the vascular bundles. Okay, open type of vascular bundles they are having, dicot and the gymnosperms. So the vascular cambium and the cork cambium appear later in the life, and they increases the girth of the, or you can say the diameter of the stem get increases. So these are the meristem that cause the increase in the girth of the organ in which they are very active. So this is known as secondary growth of the plant, and this type of growth, you can say that this is a secondary growth. So you see the growth in the plant both the way. Primary, okay, the primary growth you can see in the case of the increase in the height, that is due to the apical meristem, okay, shoot apex and the root apex, that is the primary growth, okay. And you see the secondary growth in the case of lateral meristem. In the lateral meristem, what happened? The cambium that is present in between the vascular bundle, it divides and redivides and forms the circular way of, you can say, the cambium, and it give rise xylem and phloem inner and outer side respectively, and then they will form the one of the ring in the case of new ring in the case of the stem region, and that shows the growth, and this type of growth is called as secondary growth. So here you can see, you can see here the shoot apical meristem. So it is showing the growth in the shoot apex, and there is a root apical meristem. So it is also showing the growth at the uh, apex of the root. Now there you can see in the case of lateral meristem. So this line is showing the lateral meristem, and this is present in the vascular bundles. So you can see these are present in the vascular bundle. Vascular cambium is present inside the vascular bundles, right? So the vascular bundles, uh, it the cambium that is present in the vascular bundle, it divide and redivide and forms the new layers of the cells. That increases the girth of the shoot or the root. Okay. So growth is measurable. You can measure the growth. Okay, you can measure the growth. How? The growth at a cellular level is principally a consequence of increase in the amount of the protoplasm. So what you see, what exactly you say that the growth is there at cellular level when you just checking a cell. Okay, earlier you have seen the cell was small and it is having a very limited protoplasm. Okay, now after time t, what you see, the size of the cell has been increased. The content of the protoplasm, which was there earlier, very small amount of protoplasm, now it has been increased. So the amount of protoplasm is increased now. Since increase in protoplasm is difficult to measure directly, okay, increase in protoplasm is difficult to measure directly. You cannot measure it directly. One generally measures some quantity which is more or less proportional to it. Okay, so you can measure the quantity which is more or less proportional to it. You can measure the amount of the protoplasm. So the growth is therefore measured by the variety of parameters. There is just not a single parameter that is used to measure the growth. There are variety of parameters on which your growth is dependent. The first thing, that is increase in the fresh weight. You can take the weight at the earlier stage when the cell was like in the form of immature. Okay, and after when the cell achieve a maturity, then you can take the weight of that cell again. So increase in the fresh weight, right? dry weight. You can also take the dry weight. Now, how to take the dry weight? Suppose you are want to know whether the plant has been increased its growth weight or not. So you can first take the fresh weight, and then you can go for the dry weight. You can make the plant dry in an autoclave, and then you can take the weight of that plant. Length. You can measure the length of the plant. This is also one of the parameters that gives you the measure of the growth in the plant. Okay, you can measure the height of the tree. You can measure the area that is covered, right? You can measure the volume and the cell number. You can even count the cell number. So all these are the different parameters to measure the growth in the plant. Okay, the growth in the plant. So you may find it amazing to know that one single maize root apical meristem can give rise to more than 17,500 new cells per hour. Okay, so this is the apical meristem. Apical meristem, and this apical meristem, one single apical meristem cell that is present at the root apex. Okay, root, that means that root apical meristem. So this root apical meristem, one single cell divide and redivide, and it can form 17,500 new cells in one just single hour, in 1 hour. Whereas the cells in an watermelon may increase in size by up to 350,000 times. The size, okay, the cells in a watermelon, it can increase in size. So how much time the size of the cell in the watermelon is increasing? 350,000. So these are some of the parameters that is used, okay, to denote that growth. If you measure the size of the cell in the watermelon, okay, then it is increasing how much time? Then it is showing the growth. If all the cells has been increased in such a size, then you can see the watermelon has been increased in its size. Similarly, the height of the plant. Now, in the former, the growth is expressed as increased in the cell number. Okay, so here you can see the 17,500 new cell has been formed in the case of maize at the root apical meristem. So this is showing the growth that is expressed in the increased in cell number. The latter express the growth as increase in cell of the size. Whereas here you can see in the case of watermelon, the size of the cell has been increased. Okay, so while the growth of a pollen tube is measured in the terms of its length, pollen tube, the pollen tube is formed when the pollen germinate on the stigma. Okay, the stigma, they fall on the stigma, that is called as the pollination. Now, after the pollination, what will happen? Okay, there are some hormones, there are some chemicals that is released from the stigma. It helps in the germination of the pollen tube. Okay, so pollen tube emerges out from the pollen grain, and this pollen tube penetrate the stigma region, and it also penetrate the style part, and it goes down, right? So while the growth of a pollen tube is measured in the terms of its length, so you can measure what is the length of the pollen tube. Okay, and an increase in the surface area denotes the growth of a dorsiventral leaf. So you can, this we have already, the activity I have already told you that if new leaf is emerging out, you can place it on a graph paper and you can take the area. This is a very, you can say the leaf has been emerged in an initial condition, right? Now, what do you have to do? After a week, you see that the size of the leaf has been increased. Okay, so again you can place that particular same leaf you have to do. You can place it on the graph paper and you can again take the area of that. You can take the area of that. So you see that there is an increase in the area. Okay, an increase in surface area, and that also denotes the growth in the dorsiventral leaf. The growth in the dorsiventral leaf. Phases of growth. We are having different phases of growth. The period of the growth is generally divided into three phases. So we can divide the three different phases of the growth, namely meristematic, elongation, and maturation. So these are the three different phases of the growth. Okay. So there is the first one, that is the meristematic, second is the elongation, and the third one is the maturation. So the first thing, let us understand this by looking at the root tip. The constantly dividing cell, both the root apex and the shoot apex, represent the meristematic phase of the growth. So this is the meristematic phase of the growth, that the root apex and the root shoot apex, it divides and redivides vigorously and increases the increase in the height of the plant. Okay, increase in the height of the plant. So that is showing the growth at the both the apex region, that is the root and the shoot apex region. So the cells in this region are rich in protoplasm and possesses large quiescent nuclei. So they are having the large conspicuous nuclei. So you can see this particular region. It was a very small earlier. Now you can see this particular region has been increase like this. So the detection of a zone of elongation by the parallel line technique, you can first draw this. Okay, and you can again, when you after a week or a time, particular time, you see that this length has been increased, so you can denote that length. Okay, the zones A, B, C, D is immediately behind the apex and elongated most. So here you can see which of the phase, which of the part of this has been elongated most. Their cell wall are primary in nature. Okay, if we talk about the meristematic tissue, then you can see that their cell walls are primary in nature. It is not having the secondary cell wall. So their cell wall is quite thin and cellulosic with the abundant proto-desmetal connection. So you can see the lot of proto-desmetal connection in between the cells. Right, so this is the characteristic of the cell, that their cell walls are primary in nature and they are cellulosic with the abundant protoplasmic connection present in between the cells. The cells proximal, okay, just next away from the tip, means at the tip there is a particular cell, and just below that tip, that is the proximal cell, okay, to the meristematic zone, represent the phase of elongation. So here, once the division of the cell has been taken place, okay, the cells that has been increased their number, okay, that undergoes the elongation phase. That is the phase of elongation. Okay, so meristematic zone just proximal to the meristematic zone, in which the number of cells has been increased by the process of division. Okay, division of the cells, because the meristematic cells are very active in division. So just very next to this meristematic cell, there is an that is the proximal to the meristematic zone, there is a phase of elongation. It means that the cells, new cells which has been formed by the activity of the meristematic cell, increases its size, elongation. Increased vacuolation, the vacuoles becomes large. The cell enlargement and the new cell wall deposition are the characteristics of the cells in this phase. So what happens actually in the case of elongation? That is the phase of elongation. What happens in the phase of elongation? So at the time of phase of elongation, you see the cells get enlarged. The cells increases in size. It means that cells get enlarged, and the new cell wall, because in the case of meristematic cell, there is just a primary cell wall. There is a primary cell wall. But what happens Okay, but what happens at the time of the elongation? The cell wall, okay, the secondary cell wall has been laid down. There is a deposition of the cellulose, and this is one of the characteristic feature of this phase, that is the elongation phase. Okay. Further away from the apex, that is the more proximal to the phase of elongation. Okay, then now we are going to more proximal to the phase of elongation. Then what happens? Okay, away from the apex, that is the more proximal to the phase of elongation, lies the portion of axis which is undergoing the phase of maturation. Okay, now the cells which has been elongated, now they undergoes to the maturation phase. The cells become matures now, and that is why it is called as undergoing to the maturation phase, right? The cells of this zone attain their maximal size in terms of wall thickening and the protoplasmic modifications. Okay, so the cells of this particular zone, okay, that is the maturation zone, attain their maximal size. The cells at this particular stage are at its maximal size, and in terms of wall thickening and protoplasmic modification. So whatever the protoplasmic modification is required, okay, that has been taken place. Okay, then you can also see the thickening of the wall. Okay, there is more thickening of the wall in the case of maturation zone, okay, or the maturation phase. So most of the tissue and the cell types you have studied earlier classes represent this phase. So whatever the cells you have studied about, okay, that is a parenchymatous cell, that is a collenchymatous cell, sclerenchymatous cell, okay, so they are the matured cells. Okay, they are having the matured cells. So that is why most of the tissue and the cell types you have studied in earlier classes represent this particular stage. You have studied about the maturation phase. Okay. Growth rate. How to measure the rate of growth? The increased growth per unit time is termed as growth rate. What is the increased per unit time? In 1 unit time, how much the growth has been measured? So that is called as growth rate. Thus, the rate of growth can be expressed mathematically. Mathematically, you can express the rate of the growth. An organism, as a part of the organism, can produce more cells in the variety of phase. Okay, in an organism, or the other part or a part of the organism can produce more cells in the variety of phase. The growth rate shows an increase that may be arithmetic or geometrical. So there are two different types of, you can say, the growth rate. You can measure the growth rate in the two different ways. The first one is the increase that is in the arithmetic or geometrical form. Okay, arithmetic or the geometrical form. So here you can see two forms. The arithmetic, A, that is the arithmetic, and B, that is showing the geometric. So here you can see in the case of geometric, one cell was there. It gave rise to two cells. Now each cell give rise to two cells. So there are 4 cells, then 8 cells, and then this number of cells. So here you see, what you deduce from this particular representation. So there is a geometric. In the case of geometric, you can see the geometry of the cell has been increasing. Okay. And in the case of the arithmetic, it means that the cell is dividing and increasing in the number. But here in the case of arithmetic, you can see a one particular type of cells is used to divide. You can see here, right? So that is the arithmetic. Now here you can see this geometric as well as the arithmetic, how it is occurring. Zygote divide. So this is a zygote cell and it divide in 2 cells. Now the 2 cells Two cells here, it can form the four cells. Okay, the geometric phase, all cells divide. So here all the cells are division taking place, and now in the arithmetic phase you can see the cells are capable of the divisions only here, and non-capable, okay, the cells lose the capability. Here you can see the different cells. But in the case of geometry, okay, you can see the all the cells are at the dividing phase only. All the cells are used for the division, whereas in the case of arithmetic you can see only one cell is dividing, and that is pushing towards the outside. Okay, so this is one of the representation of the arithmetic growth and the geometric growth. Now, in arithmetic growth, following mitotic cell division, only one daughter cell continues to divide while the other differentiates and matures, right? So this you see in the case of arithmetic growth. Okay, that is occurring in mitotic cell division. Arithmetic cell, it means that mitotic cell division, and only one daughter cell continues to divide while the other differentiates and mature. Other cells just they get differentiates and mature, but the division is taking just in one particular cell, one daughter cell, and that continues to divide and redivide. The simplest expression of the arithmetic growth is exemplified by the root elongation at a constant rate. So here you can see at the growth of the root, okay, the growth of the root, and this is one of the simplest expression, that is the growth of the root. And here you can see that the elongation at this particular stage, that is the growth in the case of roots, it is at the constant rate. Okay. Now, look at the figure here. The constant linear growth, okay, if you see the growth of the apex of the root, how the root is growing down. So at a particular interval of time, you just note down the, you can say after a week, 1 week, 2 week, 3 week, 4 week, 5, 6, and 6 week. So this is the height of the you can see the growth of the roots. So every time you see it is, okay, the growth is at the same rate. Okay, every week you see there is an equal increase in size from the previous one, right? So this is showing the constant linear growth in the case of root. So mathematically, it is expressed as LT is equal to L0 plus RT. This is one of the formula, LT. That is the length at a time T. Okay, after a time T, you see this length of the stem, length of the, root, length of the root. Okay, LT. That is the length at time T. L0, length at the time 0, at which you have first started to measure the length of the root. Okay, so that will be determined as 0, L0. But after the T interval of time, means a week, after second week, after third week, after fourth week, like this you will do, right? And so that is the, when you have just started, first time you just measure the length of the root. So that is L0. Okay. And length at time T, it means that after a time particular, you can say the first week, second week, then that will be LT. R is the growth rate. That is the growth rate that is divided by the elongation per unit time. Okay. How much time you are giving to it. Suppose here I've taken 1 week. So after 1 week, I will just measure the length of the root. So let us now see what happens in the geometrical growth. This we have seen in the case of arithmetic growth. So arithmetic growth examples you can see here now. Now what happened in the geometrical growth? In most system, the initial growth is slow. Okay, that is called as lag phase. The initial first time the growth you see, that is very, very slow, and that is also called as lag phase, and it increases rapidly thereafter. But after some time, this growth rate increases many folds, at an exponential rate. Okay, and that is called as exponential rate or the log or the exponential phase, right? So here both the progeny cells following mitotic cell division retain the ability to divide and continue to do so. So here what will happen, the progeny cells, the new cell which has been formed, okay, it will shows the following the mitotic cell division, it will divide and form the mitotic cell division and retain the ability to divide, and it will retain its ability to divide and continue to do so, right? So this is occurring in the case of, okay, in the case of uh, geometrical, okay, geometrical growth, how you measure the geometrical growth in a plant. So this is showing the two phases, that is the lag phase and the exponential or the log phase. Okay, so earlier the growth in the case of geometrical growth, you see that it is a sees type, it is not growing at a very you can say the uh, in a pace way. Okay, that is why it is called as a lag phase. The phase showing the very limited growth. Okay, but after the certain time, okay, it is showing the exponential growth, it is showing the exponential growth. So, however, with limited nutrient supply, when the supply is very, very less, nutrient supply is very less, the growth slows down, leading to the stationary phase. And after that, you see that the growth is showing the stationary phase. This is the graph is showing going to the horizontal form, and that is going to the stationary phase. And if we plot the parameter of growth against time, then we get a typical sigmoid or the S curve. Okay, there is a sigmoid or the S curve. It happens when if we plot the parameters of growth against time, you know that there are certain parameter of growth. Okay, and without those parameter, there will be no growth. So when you plot the parameters of growth against time, you will get the sigmoid type of curve. A sigmoid curve is a characteristics of the living organism. Okay, in nature, you see the sigmoid curve only. A sigmoid curve, it is a characteristic of the living organism growing in a natural environment. So this is going to in a natural environment, and it is typical for all the cells. Okay. So such type of growth, a sigmoid growth, and it is a very typical for all the cells, tissue and organ of the plant. Okay. In the case of different organs of the plant, you see this type of curve. In the case of tissue, in the case of different organisms also, you see this type of curve, right? So it is a typical for all cells, tissue and organ of a plant. You can see this type of curve in the growth of the cells, tissue and different organs of a plant. So can you think of more similar examples? What kind of curve you can expect in a tree showing the seasonal activities? Okay, how you see such activity when there is a seasonal growth or the seasonal activity. So here you can see an idealized sigmoid growth. In nature, you see the sigmoid growth, and that is why it is written here an idealized sigmoid growth curve. Okay, that is typical of a cells in culture and many higher plants and in the plant's organ. And this you see in the case of cell culture also. Cell culture means when you are culturing the cells in a very controlled form in a laboratory, then also you see such type of curve. Because earlier the cell divide, there is a single number of cells is very different. Suppose you have taken just one cell. So one cell divide into two. Two will be 4, then 4 to 8, 8 to 16, 16 to 32, 32 to 64. Like this, it will divide. So earlier, their number is very less. Okay, and that is why it is showing the lag phase. But the nutrient supply is very high at that particular time. If you compare with the number of cells, the nutrient is very high. And after that, what happened? The cells divide and redivide, okay, and increases its number. So this is showing the growth. But what happened? As you increase the number of cells there, the supply of the nutrient now, there is a less amount according to the growth, according to the increased number of cells. You see the less number of nutrients available now. So this is showing the exponential phase, that is the growing phase. And after that, you can see then there is a stationary phase. So this stationary phase is showing that the nutrient which is supplied in the culture medium has been reduced now. So the cells started not showing at that particular growth rate, and also the cells started dying also because of the consumption of the nutrients. So here you can see the growth now becomes stationary, right? So the exponential growth can be expressed as, so here we can see there is an exponential growth. So if you want to measure the exponential growth, you can express it as W1 is equal to W0 e to the power R into T, where W1 is the final size, okay, final size of the plant, or any you can see the size of the cell, okay. So whatever the thing you are taking in the consideration, the parameter that is, you have taken either weight or height or number, whatever the parameter you have taken, so that is the W1, that is the final size. And W0, the initial size at the beginning of the period. So whether you have taken the size, whether you have taken the weight, height or number, whatever, so that you have taken at the initial stage as well. And R, that is showing the growth rate, T is the time of the growth, and E is the base of the natural log. So if you keep all the things in this parameter, then you can get the final size. Okay, what will be the final size? So that you can get from here. Here R is the relative growth rate. Okay, R is showing the relative growth rate, and it is also the measure of the ability of the plant to produce a new plant material. So it is showing that the plant is able to, okay. So R is showing the relative growth, and it is also measured. So this R is used to measure the ability, okay, the ability to produce new plant material, how it is going to produce the new plant material. And that is referred to as efficiency index. Okay, that is showing the efficiency index. And hence the final size of W1. Okay, what will be the final size? That is the final size at the last stage when you are measuring. The final size of W1 depends upon the initial size, what you have taken as the initial size, that is the W0. Quantitative comparison between the growth of the living system can also be made in two ways. In this, you can say that it can be measured in two ways. What are those two ways? Okay, what are those two ways? First, measure and the comparison of measurement and the comparison of total growth per unit time is called as absolute growth rate. Okay, the first thing that you can measure it in a form of absolute growth rate. So what is that growth rate? Measurement and comparison of the total growth per unit time. Okay, so you are considering the total growth of the plant as per unit time, and that is called as absolute growth rate. Now, the growth of the given system per unit time expressed on a common basis. Okay, the growth of the given system per unit time, okay, expressed on a common basis, that is per unit initial parameter, and it is called as relative growth. Okay, so all the things when you are taking in the consideration, and it is called as absolute growth rate. And if you are taking it as the initial parameters, then you are comparing it. You are comparing it, and that is called as relative growth rate. So in this way, two ways you can measure the growth. That is the absolute growth rate and the relative growth rate, right? So here you can see figure 13.7. Two leaves A and B, okay, are drawn, and that are of the different sizes but show absolute increase in area in the given time to the given give leaves A1 and B1. So here you can see leaves, that is 5 centimeters square, 5 centimeters square. And you can, after a particular time period, you note down or draw its again the size, that is 10 centimeters square after a time t. So A1, okay, two leaves A and B earlier you have taken. So this leaves also you have taken, its area was 50 centimeters square, 50 centimeters square. Okay, this is 5 centimeter and this is the 50 centimeter, A and B. Okay, now absolute increase in area in the given time to give leaves. So after a time t, you have noted down its area. So it is A1, that is 10 centimeters square, and this is B1. So this is showing 55. So earlier it was 50, now it is 55. Earlier it was 5 centimeters square, now it is showing the 10 centimeters square, 10 centimeters square. So what is the difference? In the case of first, you see there is a 5 centimeters square. And in the case of second, then also you see the 5 centimeters square. So both the leaves A and B have increased their area by 5 centimeters square in a given time to produce A1 and B1 leaves, right? So, condition for growth. So, why do you not try to write down what you think are necessary, the condition for growth? So, what you can say that here in this case, what are the different conditions that are necessary for a growth? What are the conditions that are necessary for the growth? So, this list may have water, oxygen, and nutrient as very essential elements for the growth, right? So, these are very, very essential. So first of all, the water is a very important factor for the growth. The oxygen is also very important, and the other nutrients. Basically, the nutrient that plant require, it's coming from the air, water, and soil, right? So all the elements that is required for the growth, these are the things. The plant cell grow in size by the cell enlargement. Okay, the plant cell grow in size by the, if the size of the cell get enlarged, so that you can say that the size of the cell get enlarged, which in turn requires water. Okay, so the water will be absorbed by them. So the turgidity of the cells help in extension growth. So how the cells become turgid? When it absorbs more and more amount of water. Okay, the water get filled inside the cell and the cell become very turgid. So it is showing, okay, it is showing that the cells become turgid, and turgidity of the cell help in expansion of the growth. So this is showing the cells has been expanded. Just because the more and more water get filled, the cell become turgid, the cell is expanded now. Thus, plant growth and further development is intimately linked to the water status of the plant. So the water is one of the very, very important factor, right? So water also provide the medium for enzymatic activities needed for the growth. Okay, so there are so many enzymes that is required for the growth. There are so many enzymes, and these enzymes become available when there is water. Okay, so water also provide the medium for the enzyme. The enzyme is there, but there is no medium. Then how the enzyme will show its activity? How it going to increase the rate of reaction for which it is responsible? Okay, so it will not happen unless there is a medium, and the medium that is in the form of water. So oxygen help in releasing metabolic energy essential for the growth activities. Okay, so oxygen, you all know that for the cellular respiration there is an requirement of the oxygen. So oxygen help in releasing the metabolic energy that is in the form of ATP that is released at the process, you can say that is the uh, after the cellular respiration. Nutrients, there are so many nutrients that are required for the plants. Macronutrients and micronutrient. Macronutrient, the nutrients that are required by the plants in a large amount. Okay, so that is called as macronutrients. And there are certain nutrients that are required in a very small quantity. The quantity is very, very less. So that is said that it is required in a very small quantity. But it doesn't mean that they are not so important. Okay, the requirement is in a very small quantity, but their essentiality is at the same level. That is up to the macro level. Okay, it is in the same way it is important as it is important for the macro nutrients, and are required by the plant for the synthesis of protoplasm and act as a source of energy. So these nutrients are very, very important. Without it, okay, the protoplasm will not get synthesized. So you all know that protoplasm is the living component of the cell. In addition, every plant organism has an optimum temperature range that best studied for its growth. Okay, because you all know that there are so many enzymes that is required for the growth of the plant, and these enzymes are working at a particular temperature, right? So in addition, right, if you are talking about the nutrients, if you are talking about the oxygen, if you are talking about the plants, then in the same way the temperature also play an important role, okay, in the growth of the plant. And that is why, in addition, every plant organism has an optimum temperature range best suited for its growth. Any deviation from this range could be detrimental to its survival. So if the temperature is not as suitable, okay, the temperature is more or less, then the plant cannot survive, because the enzymatic reaction that is taking place will not happen. Okay, these enzymes will not be available. Environmental signals such as light and gravity also affect the certain stages of the growth. Okay, there are different environmental signal, that is light, gravity, that also affect some of the phases, or you can say the stages of the growth. Okay, so that also get affected if all these environmental signal is not available properly, then the growth is affected. So here we can see that there are so many things that are affecting the growth of the plant. The water is there, the oxygen is there, nutrients is there, okay, and certain environmental factors are also there, right? So this is all about the growth of the plant, and we have completed this particular session. In the next session we will see the hormonal activity that we see in the case of the plant growth and the development. Okay, so thank you very much for attending today's session. We will meet with another session. Till then, take care and bye.